Welcome back to Biomindscape. Today we are going to know about DNA binding motif. DNA binding proteins such as the transcription factor, it recognize and bind a short nucleotide sequence, usually complementary, which is between the protein and the surface of the double helix in the region of the binding. So how DNA binding protein can make the sequence specific? Contacts to the DS, that is double stranded DNA molecule, without breaking the hydrogen bond that hold the base together. So the contact occur between the DNA binding protein and the edges of the base pair that are exposed in the groove of the DNA, especially the major groove. Hydrogen bond provide the sequence specific contact. The amino acid of protein makes specific hydrogen bond, that is Ta base pair is different from that of the Cg base pair, that is cytosine and guanosine base pairs. So DNA binding motif permits specific binding of the DNA to many different DNA binding protein with quite different specificities, including the leucine zipper, helix loop helix, helix turn helix, and zinc finger motif. So let's see the structure of the helix turn helix. So it is like a stand is present. Here there is COOH and here there is NH2, and this is the helix. We call it as recognition helix. Recognition helix. So this is the structure of the helix turn helix motif, and the C terminal alpha helix, it is known as the recognition uh, helix because it participates in the sequence specific recognition of the DNA, and this helix fits into the major groove of the DNA. So the helix turn helix, that is the HTH. This HTH motif is present in mostly the prokaryotic and eukaryotic DNA binding protein, and this was the first DNA binding structure to be identified. It is made up of alpha helix separated by beta turn, which is made up of 4 amino acid. So the second of which is usually glycine. So this turn in the conjugation with the first alpha helix position and the second alpha helix on the surface of the protein is an orientation that enables it to fit inside the major group of the DNA molecule. So this second alpha helix, it is known as the recognition helix. We have seen this where COH is present. Above it there is a recognition helix. So the HTH, that is the helix turn helix structure, it is usually 20 or so amino acid in length, and so is just a small part of the protein as a whole. So the homeodomain, it is an extended helix turn helix motif. It is possessed by the protein which is coded by the homeotic gene. So it is made up of highly conserved domain of 60 amino acid encoded by the 180 base pair DNA sequence. We call it as an home box. So it has 3 alpha helix. The home box have 3 alpha helix, and helix 2 and 3 are separated by the beta turn, and helix 3, which is 17 amino acid residue long, act as a recognition helix and bind the major groove of DNA. So helix 1 make the contact with the minor groove. Now what is a helix loop helix motif? So let's know about the helix loop helix motif. What is it? It is different from that of the helix turn helix motif. Mostly people confuse with the helix turn helix motif and helix loop helix motif. The helix loop helix motif, it is involved in the DNA binding as well as the protein dimerization. It is composed of the two region alpha helix, which is having 15 to 16 residue. They are separated by length or variable length, and they form a loop between the two alpha helix and the length of the connecting loop, it can differ from 12 to 28 amino acid residue. So this motif is similar to that of the leucine zipper motif. And we can see in this diagram, it is given that there is an active HLH, that is helix loop helix motif homodimer, and this motif it is responsible for both dimerization and DNA binding. And how it is similar to that of the leucine zipper motif, let's see. So leucine zipper motif, this one is the leucine zipper motif. It mediate both the DNA binding and dimerization. There are two distinct right-handed alpha helix. They participate forming the homo as well as the heterodimer structure. There is a N-terminal DNA binding domain and it have positively charged amino acid residue. We can see in the diagram there are two positive charge which are present on the DNA binding domain, and there is a dimerization domain. It have all the leucine rich residues. There is a C-terminal dimerization domain. It is made up of 30 to 40 amino acid, and we know that it have leucine rich residue at every seventh position. We can see the C terminal dimerization domain, it have 30 to 40 amino acid with the leucine at every seventh position, and there are two alpha helix, they are parallel and they form a coil structure. They are wind around each other and they form this coil structure. So the leucine residue, it have the R group, which is from the alpha helix domain in which the leucine residue it is present. And there is a stabilizing homo and heterodimer. The leucine zipper domain, it is present in the DNA binding, in mainly in the DNA binding protein such as the C, juice, phos, etc. There are various names for this leucine zipper motif. Then zinc finger motif. What is a zinc finger motif? There is a zinc coordinated DNA binding motif that is called a zinc because it has the zinc group in the motif. Therefore, it is known as the zinc finger motif. There are several type of zinc finger motif, finger structure, and this, for example, we can see this is an example. Here we can see the zinc is present in between and they are attached by the circular bead like structure, and here there is a histidine domain, then various type of domains are present. Here also there is, and zinc is attached in between, and above this also there are various bead like structure. So there is C2H2 zinc finger. It is mostly DNA binding motif and this motif has 23 to 26 residue, and they are conserved by cysteine and two conservative histidine residue. There is a side chain which bind one zinc ion, and the name zinc finger was coined because the two dimension diagram of the structure it resemble a finger like structure. So when the dimensional structure was solved, it became clear that the binding of the zinc ion by the two cysteine and two histidine residue it folds the relatively shorter polypeptide sequence into a compact domain. So there's a formation of compact lobular domain in which the zinc ion it is present, and there's a tetrahedrally coordinated by the tetrahedrally cysteine and histidine residue. So zinc is essential for correct folding and DNA binding. By NMR or X-ray study, it shows that the C2 and H2 finger it is compact lobular domain. It is composed of 12 residue alpha helix which is packed again. It is packed again the irregular beta sheet with the zinc in between is present. There is a alpha helix of each finger lies in a major groove and contacts the three base pair. The base contact are almost entirely with one DNA strand. Now we have seen all the three types of the transcription regulatory protein that contain various binding motifs. So first one was the helix turn helix. It was seen in which organism? In E. coli, phage, and zinc finger. It was present in East mammal and xenopus, whereas the leucine zipper it is only found in the mammals. There are various regulatory protein which helps in this binding of the which we can see this binding like helix turn helix in lac repressor cap crow, whereas the zinc finger we can see in gal4 and tf3a, and in mammals we can see in receptor of steroid hormones, whereas in xenopus tf3a is the regulatory protein, and in yeast the gal4 is the regulatory protein, and in leucine zipper the phos-june cre binding protein, then c-mic and cebp. These are the types of regulatory protein which have this binding motif, that is the leucine zipper motif, and in it is found in the mammals. So these were the various type of the motifs. These all are the examples of the transcription regulatory proteins.